Welcome to Science with Tal. In today's discussion, we will discuss the basic principles of the olfactory system and how it gives rise to the sense of smell. We will cover the different stages of the olfactory pathway, starting from the initial sensory transduction to the different divisions of the brain that process this information. To understand how the olfactory system operates, let's begin our discussion in the region of the nasal cavity to get a good overview of the major olfactory structures. To give you some orientation about where we are, this is a sagittal cut where you can see and recognize the tongue, the hard palate, and the nasal cavity. At the top of the nasal cavity, we can find the external segments of the olfactory receptor neurons, or simply ORNs, which are the very important neurons that allow transduction of chemical odorants into neural signals that eventually allow us to process smells. The cell bodies of olfactory receptor neurons are embedded in a specific layer of cells called the olfactory epithelium, which, in addition to the ORNs, have other types of supporting cells that contribute in maintaining a healthy environment. We will shortly see what these other cell types are and what they do. ORNs generally have two functional segments to their body, one that goes into the nasal cavity to transduce odors into neural signals, and one that passes through the cribriform plate to reach the olfactory bulb. In the olfactory bulb, this is where the signals from the nasal cavity are first integrated before being sent to the different brain structures via the olfactory tract. As a last little detail about this general overview, I want to mention that a collection of olfactory receptor neurons that pass through the cribriform plate form the olfactory nerve, which is also known as cranial nerve 1. Now, if we consider a small segment of the olfactory epithelium and olfactory bulb, we can see in much greater detail what is going on. The first main component that I want to discuss is the olfactory epithelium, which, as you might remember, is the top layer of cells before we reach the cribriform plate. The olfactory epithelium is part of the nasal mucosa, which is critical for the immune protection against toxic volatile chemicals and in the humidification and warming of inhaled air. In addition to the olfactory epithelium, the nasal mucosa is also made of the respiratory epithelium, which covers some other regions of the nose that are less concerned with smell, so we will not discuss that epithelium in too much detail in this video. Besides the olfactory receptor neurons, which you can see here colored in blue, red, and green, the olfactory epithelium is also made out of Bowman's glands, basal cells, and other diverse supporting cells. Bowman's glands are a key structure in the olfactory epithelium that secrete mucus into the nasal cavity to produce a barrier that protects the cells exposed to the exterior and thus are vulnerable to toxic chemicals. The mucus produced by Bowman's glands also has the important function of trapping and dissolving odorants, which aids tremendously in our ability to smell different chemicals. As for the other two types of cells, basal cells are stem cells, which means that they are the progenitors to the different cells of the olfactory epithelium. Given that a lot of these cells are exposed to the exterior and get damaged fast, there is a high turnover rate in the olfactory epithelium that requires constant replacement, and that demand is met by the basal cells. Finally, supporting cells include different types of cells that play various roles for the proper health and functioning of the olfactory epithelium. Now, let's cover in more detail the structure and function of olfactory receptor neurons. As I said earlier, these neurons have two general functional units, one that goes in the nasal cavity and converts smells to neural signals, and one that transmits the signals to brain structures. This functional division is made possible mainly because ORNs are bipolar neurons. The end that is in contact with the mucus layer and the odorants is also referred to as the apical end and contains the specialized olfactory cilia. These cilia are immotile and are analogous to the typical dendrites of neurons. It is in this region that the sensory transduction from chemical to electrical signal will happen. The end of the ORNs that goes towards the brain is the basal end, and it is essentially the axon of the neuron. An important point to add as well is that this basal end or axon is unmyelinated, but that is not too big of a concern for these neurons because the distances they have to carry the information over is rather short. Now, before we cover what signal is sent to the brain, we first have to understand how different odorants are transduced into neural signals. To do so, we have to take a closer look at the membrane of a given olfactory cilia. It turns out that the main mechanism that allows to transduce odors is mediated by G protein coupled receptors, or simply GPCRs. As a quick note, This section about the sensory transduction will involve a few different molecular mechanisms that I have covered in more detail in other videos, so if these terms are new for you, I suggest watching these videos about GPCRs and different kinases to understand the broader context and get familiar with the terminology involved with these proteins. Now, at the membrane of the ORN, we can first find the G protein-coupled odorant receptor and an inactive G protein. The first step that activates the transduction pathway is the presence of the odorant, which will be perceived by its ability to bind to its corresponding receptor. The binding of the odorant will activate and change the conformation of the receptor, which in return will be able to activate the G protein. The activation of the G protein will switch the GDP of the alpha subunit to GTP and cause the dissociation of the alpha subunit from the beta and gamma complex. The activated alpha subunit then goes on to activate the olfactory specific adenylyl cyclase 3, or simply AC3, and when active, AC3 will convert ATPs to cyclic AMPs. The newly made cyclic AMPs are then able to bind to cyclic AMP-gated channels and open them, which lets sodium and calcium flow inside the cell. The entry of these two positively charged ionic species constitutes the first depolarization for the olfactory receptor neurons. Now, at this point, the neuron is slightly depolarized, but the signal is not strong enough to cause an action potential. The bulk of the depolarization that will cause the neurons to fire an action potential actually comes from the opening of calcium-gated chloride channels by the binding of calcium. When this channel opens, chloride leaves the cell, and since it is negatively charged, it will depolarize the cell. One thing to keep in mind with these ORNs is that they do not have the same concentration gradients as typical neurons have. Indeed, ORNs don't pump chloride outside of their body, so they hold a lot of that chloride inside, which means that when the chloride channel opens, the chloride exits the cell instead of entering inside as it does in typical neurons. If a sufficient amount of sodium and calcium enter and chloride exits the cell, an action potential will be fired and will communicate to the rest of the nervous system that an odor is present. The steps circled in white, from 1 to 8, are generally considered to be the steps that lead to a depolarization and an eventual action potential, but there is much more to consider about this pathway. Namely, we also have to consider the steps of the pathway that lead to the recovery from the depolarization and the adaptation to the signal. To be more exact, adaptation is the process by which the sensitivity to an ongoing stimuli is modulated. A good example of adaptation that happens quite often in our lives is when we visit a place for the first time, and at first we find that this place has a particular smell. But after a while of being in that place, you will probably notice that this smell dissipates, although the same odorants that make up the smell are presumably still there. This phenomenon is explained by the process of adaptation. I bundled the steps for adaptation and recovery together because it is still unclear which steps contribute to either recovery or adaptation. Now, the first step of recovery and adaptation is the removal of calcium at the membrane by specific transporters. There are two known transporters that can be used by ORNs, the NCX and NKCX transporters. The difference between these two is which ions are used to move calcium. The NCX uses the gradient of sodium to remove calcium, and the NKCX uses the gradient of potassium and sodium. The removal of calcium along with the other positive ions causes a net hyperpolarization in the cell, which contributes to recovering from the depolarization. Now, the reason why the removal of calcium is so important is because in addition to opening chloride channels, calcium is the catalyst to a bunch of different molecular pathways that play a role in recovery and adaptation. To do so, calcium can form a complex with calmodulin. Then, this complex can first inhibit or desensitize the cyclic AMP channels to cAMP, which will reduce the amount of calcium that enters. Second, the complex can activate a phosphodiesterase that will convert cyclic AMP to AMP, which again will decrease the amount of depolarization produced because AMP cannot open cyclic AMP-gated channels. The third possibility for the calcium-calmodulin complex is to activate the calcium-calmodulin-dependent kinase type 2, or simply CAMK2, which upon activation can phosphorylate AC3, and that phosphorylation will inhibit its activity. So as you can see, calcium has many effects in this pathway, and for that reason it is highly regulated. In addition to calcium, another important chemical that you can see being heavily regulated is cyclic AMP. Beyond activating the cyclic AMP-gated channels, a sufficient amount of cAMP can also activate protein kinase A or PKA, and with the contribution from another protein called the G protein receptor kinase or GRK, active PKA and GRK can phosphorylate the GPCOR. This phosphorylation triggers the involvement of beta-arrestin and other adapter proteins, which essentially cause the GPCR to get capped off. The capping off will result in the signal being inhibited because if you remember from step 2 in the depolarization steps, it is the conformational change of the GPCR that allows to initiate the entire pathway. Finally, another important step that contributes to recovery and adaptation is the hydrolysis of GTP of the alpha subunit to GDP by RGS2. This event contributes to the reformation of the G protein and the inactivation of the signal. As you can see, this transduction mechanism pathway is very complex, but the crazy thing is that a lot of it remains to be discovered, especially when it comes to the adaptation mechanisms. Now, before we go back to the olfactory epithelium, there are a few properties about ORNs and odorants that I want to discuss because they are very important in the way we process smells. So, it turns out that in our genome, we have about 400 functional genes that code for different olfactory receptors, so it is a very diverse family of genes. Another important fact is that most ORNs express only one odorant receptor gene. So, for example, you might remember that in the olfactory epithelium, we had blue, red, and green ORNs. Well, for each of these ORNs, the main difference maker is that the blue ORN, which we will call ORN A, expresses the olfactory receptor A, but the red ORN, or ORN B, expresses the olfactory receptor B. Same thing goes for the green ORN or ORN C. So, more simply put, blue ORNs have blue receptors, red ORNs have red receptors, and green ORNs have green receptors. Now, although for the most part there is one type of receptor per ORN, each receptor can be responsive to different odorants. That means that, for example, each receptor is responsive to their own respective color of odorant, but in addition to that, odorant receptor A and C can also detect the yellow odorant. As you can see, one receptor can detect multiple smells, but you will also notice that one type of odorant can bind to multiple receptors, as the yellow odorant illustrates. As such, if we consider each odorant and which ORN responds to it, each odorant will be able to produce a different perceived smell due to the overlap in odorant receptors and odorants. This form of interpretation for a sense is also known more generally as combinatorial processing, which puts forward the idea that each distinct smell is encoded by a unique combination of neurons. So even though the yellow odorant D can bind to the same receptor as the blue odorant A, its smell is encoded differently because odorant D can also bind to receptor C. If this is the first time you are covering this topic, make sure to go slowly through these ideas because I know how confusing it can be at first to get how the system works. Now, tied to this idea of combinatorial processing, I also want to talk about two factors that influence the processing of odorants. The first factor is obviously the chemical composition of the odorant. A subtle and often utilized example to showcase this property is the difference in smell between two enantiomers, R and S carvone. Enantiomers are pairs of molecules that act essentially as mirror images of each other, and for that reason, they can't be stacked on top of each other, which makes them two different compounds. As such, R carvone has been noted to smell like spearmint, whereas S carvone smells like caraway seeds. This difference in smell can simply be explained by the fact that the two structures of the odorants directly influence the binding site for which receptor they will bind to. The second factor that impacts the perceived smell is the concentration of the odorant. A good example of this is thioterpeneol. At low concentration, this odorant has a fruity smell, but when its concentration becomes high, this compound starts to smell putrid. This can be explained by the fact that olfactory receptors have different affinities to different compounds, and accordingly, it might take a very high concentration of a given odorant to activate that olfactory receptor. As a result, this dependence on the concentration can directly change the code and result in different smells. These few bits of information should be sufficient for us to understand what happens at the level of the olfactory epithelium. Now back to the schematic. Now that we have covered the transduction mechanism and combinatorial processing, we are well equipped to continue our discussion in this diagram. So, on the basal side of the olfactory neurons, the axons leave and pass through the cribriform plate to reach the olfactory bulb, which is represented by this large yellow layer. It turns out that the olfactory bulb can be divided into 5 different layers that each process a specific function. The most external layer of the olfactory bulb is named the glomerular cell layer, and it is characterized by the fact that it contains structures known as glomeruli or glomerulus in singular. In the diagram, each glomerulus is represented by a round circle, and that is because a glomerulus is simply accumulations of neuropil, which is simply a dense network of synapse and glial cells. The synapses in question that are important to consider are the connections between the axons of the olfactory neurons and the dendrites of projection neurons that relay the signal to the brain. An important aspect that you will notice in this diagram is that each glomerulus processes information for one subtype of ORN. Hence, all blue ORNs, which all express the same receptor, connect to the same glomerulus, and the same goes for the red and green glomeruli. This is another example of how the idea of combinatorial processing is exemplified in the olfactory system. In addition to the synapses between the ORNs and the projection neurons, each glomerulus also contains the input from periglomerular cells, which are simply interneurons that modulate the activity inside the glomerulus. Now, in terms of the projection neurons, there are 2 main types that we can consider, and the 2 are located in 2 different layers. The principal type of projection neuron is the mitral cell, and its cell body is located in the mitral cell layer. The other type of projection neuron is the tufted cell, and is located in the external plexiform layer. As I said, projection neurons integrate the signals from ORNs and send them to the brain. The most internal layer of the olfactory bulb is called the granule layer and contains a particular type of interneuron called the granule cell. Here again, granule cells offer some modulatory mechanisms to regulate the activity of projection neurons. The last layer is the internal plexiform layer, which relative to the other layers is not too interesting as it does not contain any cell bodies but mostly axons from different cells. This final detail completes our picture on the mechanisms and different cell types in the peripheral structures of the olfactory system. We are now ready to move on to what happens in the brain, and to do so, I would like to consider a ventral or bottom view of the brain to best see the regions involved in processing smells. Before we start, I want to make evident to you that much remains to be discovered about the different cortical areas associated with olfaction, and as such, what I explained reflects what is generally understood today. As a reminder of where we are, here you can see the olfactory bulb, which is where the signals from the ORNs were integrated and then sent through the olfactory tract. As the olfactory tract reaches the region of the optic chiasm, it divides into the lateral, intermediate, and medial olfactory stria. Each stria then sends olfactory information to different cortical areas. The main projection area for the olfactory tract is known as the piriform cortex and corresponds essentially to the primary olfactory cortex. In terms of its function, the piriform cortex is believed to process the odors based on their identity and intensity without taking into consideration their value. A noteworthy property of the piriform cortex is that it is made out of 3 layers, which puts this region to be very evolutionarily ancient in comparison to the typical 6-layered neocortex that makes up the majority of our brain. Another area important in the identification of odors is the entorhinal cortex. In patients that have Alzheimer's disease, this region of the brain sometimes degenerates and leads to an associated loss of being able to tell which odor is smelled, so this region presumably plays a part in the identification of odors. Downstream of the piriform cortex, a very important area involved in olfaction is the orbitofrontal cortex. This area receives projections from the piriform cortex and many other senses such as vision, touch, and taste, and it integrates all of that together to form complex multimodal representations of our surroundings. It is in that area where the value or pleasantness of an odor will be processed, as well as the ability to compare and discriminate odors from each other. The last 2 structures that I feel are noteworthy to mention here are the anterior olfactory nucleus, which can be found in the anterior portion of the olfactory tract, and the olfactory tubercle, which is located at the end of the intermediate stria. The interior olfactory nucleus is responsible for providing connections between the 2 olfactory bulbs via the interior commissure, and is also responsible for sending reciprocal connections to the ipsilateral olfactory bulb. These reciprocal connections are believed to modulate from the cortex what happens inside the olfactory bulb. When it comes to the olfactory tubercle, it receives projections from the olfactory bulb, and it plays a role in the reward system. As such, that structure integrates many senses together. Finally, To complete our discussion on the olfactory cortex, there are a few more structures that have been shown to be important in olfaction. These remaining structures cannot be seen from the exterior surface of the brain and need to be seen from various angles. To orient them, we can first consider this mid-sagittal or medial view of the brain. Under this view, there are two important structures, namely the thalamus and hypothalamus. If you are unfamiliar with the thalamus, simply put, the thalamus is a collection of nuclei that receive and project information throughout the entire brain. In particular, it receives projections from the piriform cortex to the mediodorsal nucleus, which then sends that information to the frontal lobe, including the orbitofrontal cortex. These connections are believed to play a role in olfactory-guided memory. As for the hypothalamus, it is also a collection of nuclei, and it is an important region for our autonomic behaviors. With respect to olfaction, it mediates the involuntary reactions our body produce, such as salivation and gastric activity. To see the next structure, which is the amygdala, we need to take an anterior cross-section at this level, and much like the thalamus and the hypothalamus, the amygdala is also a collection of nuclei. In terms of its involvement in olfaction, it has reciprocal connections with the piriform cortex, the orbital frontal cortex, and the thalamus, and it is believed to play a role in complex cognitive functions such as the storing of odor value and learning. As a quick note about this cross-section, you can see at this level the anterior commissure, which was important for the roles of the anterior olfactory nucleus. The last structure, the hippocampus, can be found at a more posterior level, and it is believed to play a crucial role in olfactory-guided memory from the projections it receives from the piriform cortex. Unfortunately, much remains to be discovered about the role of the hippocampus. And with this being said, this last region concludes our discussion on the olfactory system. In this general overview, you can see the different diagrams that we have covered throughout this video and how they relate to each other to mediate the sense of smell. Thank you for watching this video. If there was anything unclear or there was a mistake somewhere in the video, make sure to let me know in the comment section. If you enjoyed this video and found it useful, you can consider leaving a like and subscribing to support the channel. Thank you again for watching, and I'll see you in our next discussion.